Hello friends, myself Professor Shantaya Gurmat, Department of Botany, MGVC Arts, Commerce and Science College, Muddebiyal, Karnataka State. Friends, today's class we discuss about Pteridophytes. The best example is Equisetum. Equisetum comes under divisions Spinophyta, class Spinophylopsida, order Equisetaceae, family Equisetaceae, the genus Equisetum. Then distribution. 25 species of Equisetum are widely distributed and reach luxuriant development in the north temperate zone. Some occurs in tropical regions in Arctic and alpine zones. Then we discuss about external structure. The plant body of Equisetum represents the sporophyte generation and is differentiated root, stem and leaves. First we discuss about stem. The main stem is extensively branched, sympodial, dark brown, rhizome that gives off branches from its nodes. The rhizome penetrates deep into the soil and gives off aerial as well as subterranean branches that alternates with the brown scale leaves. The leaves are fused at their bases and along a portion of their length near its base and grow out as branches that may be remain underground or become aerial branches. They are conspicuously jointed and are green, rough, abrasive due to deposits of silica, and stiff and erect in thick and short forms. The jointed appearance is due to the appearance of distinct nodes and short forms. Nodes bear whorled leaves that are laterally fused at base. The internodes are long and hollow and are longitudinally ridged and grooved. The ridges alternate in successive internodes and so do the grooves. The number of ridges corresponds to the number of leaves at a node. Then we discuss about leaves. Equisetum is microphyllous. The leaves are simple, ligulate, isophilous, and extremely reduced in size. They arise in whorls in each node, and their number varies with species or even in the same species. Then we discuss about roots. Roots are generally born on the rhizomes and except the first root of the embryo. The rest are all adventitious and arise endogenously. Then we discuss about the internal structure. First one is internal structure of stem. Transverse section of Equisetum stem shows three important regions: epidermis, cortex and stila. First we know about the epidermis. It is a single layered tissue of the stem forming the strong outer covering. Their walls are cuticularized and covered with rods or grains of silica. The silica strengthens the cell wall, prevents water or loss through epidermis, protects against pathogens and predators, and help in keeping the plant erect. Then stomata. Stomata are peculiar in having a double set of cells lying on the above the other. The innermost cells constituting a pair of guard cells, the subsidiary cells completely cover the guard cells, and the two uppermost a pair of accessory or subsidiary cells. Then we discuss about cortex. The cortex can be divided into two parts, outer and inner cortex. First we know about outer cortex. Outer cortex is differentiated into two types of cells, sclerenchymatous and otherwise chlorenchymatous. First we know about sclerenchymatous. These are thick-walled cells that give mechanical strength to the stem and are subepidermal in position. Below the ridges, they occur in large and heavy groups. There is an equal number of smaller groups of sclerenchyma below the epidermis of the grooves or furrows. Then chlorenchyma cells. A chlorenchyma cells means a parenchyma cells contains chloroplast. These are lying lateral and below this sclerenchyma and contain a large number of chloroplasts in their cells and form assimilatory or photosynthetic region of the stem. Next, inner cortex. It consists of a few layers of larger celled parenchyma traversed by a ring of large are called vallicular canals. Each of these lies below the furrow and the external surface and is thus closed beneath the photosynthetic tissue. Then endodermis. A variety of conditions is found as to the position of endodermis in various species. Next is stelae. Within the endodermis, there is a single layer of pericycle forming the outermost layer of the stelae. The stelae consists of well-defined ring of a small vascular bundles surrounding a large pith cavity. The vascular bundles are conjoined, collateral, and endarch. They alternate in position with the vallicular canals and so lie beneath the ridges. The individual bundles in a ring are separated from one another by a large zone of parenchyma. The internal to the vascular bundles and so beneath the ridges are smaller carinal cavities. One of these belongs to each vascular bundle and is filled with water. The vascular bundles consist of xylem and phloem. There is no cambium, hence the bundles are closed. Then TS of rhizome. It differs from the aerial stem in certain respects. There are no stomata in the epidermis. The sclerenchyma is not very well developed and forms a continuous layer. Chlorenchyma is absent. The pith is present, some of species, but absent in others. Then the most important one is we discuss about reproduction by spores. Equisetum is homosporous. The sporangia are born on sporangiophores, which are very different form the ordinary leaves and are grouped together in stobuli that are terminal in position. Then stobulus. Stobulus are always terminal and solitary, means single. The stobuli consists of a central thick axis which bears a number of densely crowded T-shaped peltate scales called the sporangiophores. The sporangiophores are arranged on the axis in successive whorls, alternating through somewhat irregularly with one another. The number of sporangiophores is an uncertain question. Structure of sporangiophore. The sporangiophore has two regions. First one is small proximal cylindrical and stalk-like portion attached at right angles to the cone axis. Second one is a shield-shaped peltate disc attached to the distal or outer end of the stalk. The peltate disc bears on its under surface, close to its edge, a ring of several, usually 5 to 10 sac-like pendant sporangia. They extend inwards towards the axis of the cone and fill up all the space that is left between it and the sporangiophores. Due to mutual pressure, the peltate disc usually becomes hexagonal in outline. At the base of the cone, the central axis bears a ring-like outgrowths, the annuals. Occasionally, there are two such rings. Then we discuss about sporangia. A mature sporangium is an elongated sac-like structure full of large number of haploid spores. The spores are equal in size, means homosporous in condition. The mature sporangium has a single layer of wall cells. The cell walls of this layer are spirally thickened. A young sporangium has 2 to 4 layered thick walls. The innermost layer is derived from the outermost sporangio cells, sporogenous cells, and is called the tapetum. The inner wall layers, the tapetum, disorganize as the spores are formed. At maturity, the stobulus axis elongates slightly and thus loosens its competence. The spores, when young, are green and are covered by a thin wall of cellulose At maturity, they are relatively larger, round, and contain numerous chloroplasts and have thick spore wall differentiated into four layers or outermost epiospores. Middle perispores, third layer called the exospores. Exospore and innermost endospore. The elaters help in the dispersal of spores and dehiscence of the sporangium. Then germination of spores. The haploid spore germinates into an gametophyte. They are green and remain variable from 50 to 20 days and are unable to germinate after this period. The mature gametophytes are dull, brownish green, thalloid structures generally found in abundance on the clay soil along the banks of streams and rivers. The prothalli are monoecious and bear both Antheridae and Archegonia on the same prothallus. Then Archegonium. A mature Archegonium has the base sunken in the prothallus. It is a short and consists of 4 rows and usually 3 or 4 cells in each row of the uppermost tier. The axial row consists of an egg cell and a ventral canal cell. Then Antheridium. The mature Antheridium is more or less globular structure. Jacket may consist of 3 to 5 layers. It encloses a mass of spermatozoids and water is essential for fertilization. There is the life cycle of Equisetum. Here there are 2 generations. One is the gametophytic and another one is sporophytic generation. Here the Equisetum plant itself is sporophytic in nature and it produces the strobili and it contains number of sporangiophores, contains number of sporangia. Each sporangia contains spores. Each spore mother cell by reductional division there is a formation of haploid spores or monospores. Each spore germinate to produce the that is a gametophyte and each gametophyte bears 2 kinds of sex organs, Antheridium and Archegonium. Then Antheridium produces the male gamete means the sperm and Archegonium produces the egg. The water is essential for fertilization and there is a formation of a zygote. Then each zygote there is a development of the sporophytic Equisetum plant. So this is the life cycle of Equisetum plant. Okay, thank you friends. So we meet in the next video. Thank you.